Greetings. Today we are discussing the paper Tuning the Flexibility of Glycine-Serine Linkers to Allow Rational Design of Multi-Domain Proteins by Merkx, which was published in 2017 in Biochemistry. The generation of fusion proteins consisting of multiple protein domains is a popular and highly successful approach in protein engineering. Linkers directly affect the functionalities of such proteins. The linkers are typically consisted of repeats of glycine and serine residues. The combination of flexible and hydrophilic residues in this linker prevents the formation of secondary structures and reduces the likelihood that the linkers will interfere with the folding and function of the protein domains. The two models that were used in this study are worm-like chain model and the Gaussian chain model. So the worm-like chain model describes the polypeptide as a continuous semi-flexible tube with a persistent memory of its direction. For a short or very stiff chain, the worm-like chain model describes the property of real polymers more accurately than the other model, which is called the Gaussian chain model does, while the two models are identical in the limit of long and flexible chains. The figure 2 shows the fluorescence emission spectra for the protein of our interest. Here it's denoted as CLY. As you can see, it's cyan and yellow, and it's connected with a linker, so CLY. So the figure 2 shows the fluorescence emission spectra for CLY proteins with 3 different linkers as a function of linker length. When the emission spectra are normalized at the ECFP, or the cyan emission at 475 nanometer, a clear decrease in the yellow emission at 527 nanometers is observed, with the increase in length of the linker for each series of the proteins. This is consistent with the decrease in FRET efficiency. A trend was also observed of decreasing FRET with decreasing glycine. To theoretically predict FRET efficiency for a given linker length and stiffness and enable a comparison with experimental results, the interchromophore distance, or in short, RC, was calculated from end-to-end distances. It was also observed that each end-to-end distance sampled by many linker conformations with a fluorescent protein oriented in different directions results in many different possible values of RC for each RE. The figure 3a shows that end-to-end distance probability distribution as predicted by the WLC with a contour length of 73 amino acids and different persistent lengths, which gives a numerical relation between the end-to-end distance and the average interchromophore distance. The persistent length of flexible linker, or in short LP, also determines the effective concentration of intramolecular interactions that they connect, and in this way affects the strength of their interaction. The probability of finding a given end-to-end distance can be used to calculate effective concentration, denoted as CF, for the interactive partners at a particular distance.